All right, welcome, ladies and gentlemen. Today we are going to start chapter 7, where we're going to talk about your membrane structure and function. So, really, we're focusing here on your phospholipid bilayers. So, we're talking about your plasma membrane or your cell membrane, right? And this is the boundary that separates living cells from its non living surroundings. And when we looked at evolution, this was a really big step forward in how we believe life eventually was capable of existing because we were able to wall off, cordon off chemical reactions and other processes within these membranes, which made them way more efficient than having them just floating around in the oceans. Now, remember that your plasma membrane is selectively permeable. And what we're really saying here is that, air quotes here, we're choosing what goes across it. Um, Now, I put the air quotes around that word choose because remember that there really is no intelligent decision making here. It's all going to come down to the chemistry of what's trying to move across the membrane as well as the chemistry of the membrane itself. So, that interaction between chemical properties is what's going to allow certain things to pass across that membrane relatively easily, while other things are going to need some help to get across. The big driving force between the environment and the internals of the cell really comes down to the chemistry of these phospholipids. This really is going to be the selective barrier itself. So the chemistry of these phospholipids, in addition to whatever molecule we're trying to move and its chemistry, do they interact with each other or do they not? So your phospholipids are the most abundant lipid in your plasma membrane. And remember that they're amphipathic. So remember that that term means that it has both regions that are hydrophilic and hydrophobic. So when we look at your phospholipids, right, that head has that phosphate group on there, which makes that end of the molecule polar. And remember that like dissolves like. So those polar heads, those hydrophilic heads, like to interact with the water. And as a result, they're going to end up orienting themselves to wherever that water exists. And that water exists out in the environment, whether it's your internal, like your tissue fluids and your blood, or just if you're a bacteria floating around, a protist floating around, and you have lots of water in your environment, those heads are going to orient themselves in that direction. You also have a lot of water in your cytosol inside your cell, and so you get a lot of water there as a result, and so the heads also orient themselves in that direction too. In the center, then, you have that hydrophobic layer, which is your fatty acid tails here that are part of that phospholipid structure. Those hydrophobic tails are nonpolar. And so, again, like dissolves like. So, polar water is not like nonpolar fatty acid tails. And so, those fatty acid tails then bury themselves in the center of the membrane as a way to avoid that water. Now, here's an interesting question that has come up on exams in the past. What if instead of there being water internal and external to your cell membrane, what if it were oil instead? What would that do to the structure of that cell membrane if I had oil here instead of water? Hopefully you were able to come up with the fact that this membrane would invert itself. You would end up burying those hydrophilic heads here in the center because they're polar. Oil is nonpolar. And so the fatty acid tails, which are nonpolar, would then interact with the oil on either side. So hydrophilic heads would be in the middle, fatty acid tails would be sticking out if we replace this water with oil instead. So let's talk some history here. We didn't always know what our membranes looked like. It took some study and experimentation to figure that out. And so if we look back into the early 1900s, there were a handful of models sitting around. One that was particularly popular at the time was this model called the sandwich model. And this was developed in 1935 by Davison and Denali. And this one kind of hung around for a while. And what these two gentlemen proposed was that that cell membrane did include phospholipids, did include proteins. They knew that much at this point. But this model proposed that your phospholipids sat on either side, kind of like sandwich in a bread, and scratch that, reverse it. Bread in a sandwich, your phospholipids were the bread, and your globular proteins would sit in between those phospholipid layers, much like the meat, lettuce, cheese, that kind of stuff. So the phospholipids were the bread, the proteins were the center where that meat and that cheese and that lettuce would have existed. That was what they were talking about with this sandwich model. Literally, the proteins were sandwiched in between those two layers of phospholipids. Problem is, as we continued to experiment on the phospholipid membrane, we realized that this didn't quite fit, particularly as we continued to dive into the membrane proteins and the properties of those proteins. And one of the big things we discovered is that a lot of those membrane proteins were amphipathic as well. They had both hydrophilic and hydrophobic regions, which, again, if we assume that this sandwich model were to be accurate, that would mean that all of those proteins would need to be hydrophobic across the board. That's the only way they would be able to sandwich themselves between those fatty acid tails, because those fatty acid tails are hydrophobic. So this didn't quite hold up. So 1972 comes around and a new model gets proposed by Singer and Nicholson, and they propose the fluid mosaic model. And this is still the model that we're using today. And basically what they said is that those membrane proteins had to be dispersed throughout the membrane itself. So they were individually inserted within that phospholipid bilayer as a way to account for those hydrophilic and hydrophobic regions, so that the hydrophilic regions of the proteins would have stuck out on either side of the membrane, just like the phospholipid heads, and then the hydrophobic regions would be buried inside the membrane along with those fatty acid tails. Part of their model also takes into account just the fluidity and the uh, way that these pieces are fit together. So they suggested that the model was fluid in and of itself, meaning that phospholipids had the capacity to move around, proteins had the capacity to move around. There was capabilities for flexing of those phospholipid tails to be able to move things across and through them, and that it was just a giant mosaic itself. And if you don't know what a mosaic is, pause the video and go Google it. All right, mosaics are generally pieces of art that are made from smaller pieces. So a lot of times it's broken porcelain or broken glass or um, small components that when you look at them individually don't really seem to make any sort of picture, but when you put them together and zoom out, then you have a actual image to look at. And your fluid membrane, your plasma membrane, is just that. It's a mosaic of carbohydrates and lipids and proteins and cholesterol and all sorts of components that individually may not seem particularly important, but you put them all together and they functionally allowed the cell to be selectively permeable to its exterior environment. How did we figure this out? Well, freeze fracture studies were a big supportive piece of evidence for us to understand that this fluid mosaic model did in fact make sense. And so freeze fracture studies, essentially what they do is they take that cell, they freeze it, and then they are able to create a fracture plane. And that fracture plane goes right down the middle of those fatty acid tails. There's nothing holding those fatty acid tails to each other than their attraction to the same environment. And so when you cut it with a knife, you're able to fracture it into pieces, into layers. Much like if you've ever hit things like slate, and you get those flakes of layers that come off of it. That's kind of what's happening here with that cell membrane. But what they were able to do is when they fractured those plasma membranes, they were able to see that proteins stuck in one layer or the other. And what then we saw on the opposite side of that were the holes that were left by those proteins. And so that gave us some really good evidence to suggest that those proteins had to be integrated or go straight through the membrane. They couldn't be sandwiched in between them or stuck on one side or the other for that matter. So there's definite fluidity to your membranes. Phospholipids, for example, are very capable of moving within the membrane itself. In fact, if we look at phospholipids within their own layer. They can do what we call lateral movement, so moving side to side within the same layer that they exist in. We can see that happening somewhere on the order of 10 to 7 times per second. That's a whole lot of movement going on. They can also flip-flop from one layer to the other, but that's considerably more rare. And so you see that maybe once a month at most. You've also got fluidity in the membrane based on the structure of those fatty acid tails. So typically when you see diagrams of phospholipids, they'll show you one tail that is straight, aka it's saturated, so it's got the maximum number of hydrogens possible, while the other tail kind of has that kink to it. And that kink represents that unsaturated fatty acid, because you'll get those double bonds between those carbons, so less than the max number of hydrogens. And when you do that, it changes the bond angle between those carbons, and that's where that kink comes from. So depending on how many saturated versus unsaturated fatty acid tails you have also plays a role in how fluid the membrane is. The more unsaturated, the more fluid it is. The more saturated it is, the more viscous or thick, the less it's going to move around. You also have cholesterol that's embedded within that plasma membrane as well. And keep in mind, cholesterol, you've probably heard it associated with cardiovascular disease. There's good cholesterol and bad cholesterol, your LDLs, your HDLs. And you need the good cholesterol in your body for a handful of things. One, it does serve as a precursor to a lot of our hormones, particularly our sex hormones are a big component that initially starts off as cholesterol and we modify it from there. But it's also a major component of your plasma membranes. And when it's in your plasma membrane, it acts as a temperature buffer. So it sort of prevents the really tight packing of phospholipids, which is good. So when you get really warm, we allow the membrane to not get super, super fluid, that you're just kind of oozing phospholipids everywhere and you just kind of become this puddle of ooze. But on the flip side, it also keeps them from getting too tightly packed together when you get too cold. And if we were to do that, that membrane would just go so rigid you wouldn't be able to move things across it, and that's not good either. So those cholesterol molecules help to buffer out the fluidity of that membrane at high temperatures, keeping it more stable and rigid, at lower temperatures, allowing for some additional flexibility. Now, of course, there are ranges at which that cholesterol is capable of doing that buffering. But so long as we're not within super high dangerous temperatures or super low dangerous temperatures, it's capable of maintaining that fluidity, which is a good thing. So not only do the phospholipids have the capacity to move, but so do the proteins. How do we know that? Well, there's experimental evidence to back it up. And that's what you're looking at here in this diagram. So what they ended up doing was taking two cells, one from a human, one from a mouse, and they were able to label those proteins and then induce an event where those cells hybridized. So basically throwing electric shock at them and it forces them to come together as a single cell. So they then looked at what happened to the labeled proteins after that hybrid cell was produced. And that's what gave us the evidence to know that those proteins moved. Why? Well, look at it. Here we can see those colors of those proteins moved all over the surface of this hybrid cell. That's what told us those membranes were capable, or those proteins, I should say, in the membrane were capable of moving. Think about it. What would that hybrid cell have looked like if those membrane proteins were static in their location in the membrane? If those membrane proteins weren't able to move, hopefully you were able to come up with the fact that one side of this would be pink, the other side would be purple. All right, these would all be the mouse proteins, these would all be the human proteins, and there would be no interdispersal intermixing here. But that's not what happened. We were able to see that things were all mixed together, which gave us the ability to